So following Stein's fantastic presentation, Peter and I will moderate this second panel, panel 1.2, about the challenges and opportunities in leveraging AI ML advancements for tackling cell type, brain cell atlas questions. Peter and I will first give you a very brief introduction before our panel discussions. I will talk about the key example brain cell atlas questions that can be tackled by AI ML models. And then Peter will give you an overview of the current state of the different kinds of models that can be applied to this kind of problems. Okay, so in BICAN, as you have seen, we're generating many different types of data. And our main goal is to build brain cell atlases across different modalities and across species and, of course, across time as well. So we're generating multimodal data, transcriptomics, epigenomics, spatial, and now even adding protein level data set. We're tackling the entire brain from mouse to human, and as well as spinal cord is now being added into the picture. And we are also tackling the issue of development and evolution. We're doing cross-species comparisons, and we are generating developmental cell atlases. And of course, one of the main goals is to generate a consensus, maybe not a consensus, as we just discussed, cross-species aligned cell type taxonomies. And there are also grants that study the individual variability, especially in the human populations. And within the consortium, we established, as Ed mentioned earlier, standardized pipelines and infrastructure that can be applied to community data as well. And our ultimate goal is to build this cell type knowledge base. Okay. So then beyond this, beyond BICAN, there's many community data being generated, data types being generated for much broader purposes. Like what Stein mentioned, there is tremendous amount of genomic studies and genetic variant studies for different kinds of diseases. There are omics profiling of a variety of diseases as well as functional states. And there's also very important in vitro models such as organoids and how to develop better organoids. There are perturbational functional studies, for example, Perturb-seq data, some of which, as mentioned by Xiaowei Zhuang. And then beyond this kind of omics data sets, there is also, the study about the structure and function of the brain, data about the morphology and some projections, nanoscale and mesoscale connectivity in the brain, and multimodal single cell profiling that link molecular information with anatomy, physiology, and function using methods such as patch-seq, barcoded connectomics, and things like that. And now, of course, there's, a variety of there are many different labs study the in vivo physiology and neuronal activity in animals under different kinds of behavior conditions. So the models that, our goal is to leverage the brain cell atlas as foundations to be able to integrate and understand all this variety of community data. And if we build models to better understand the brain cell atlas data, there's also the opportunity to expand the knowledge that we gained here into this vast variety of applications in the community. And that is really our goal here, setting the foundation for the community, for the field. So of course, our ultimate goal is to build this brain cell knowledge base that can bridge information at the genes and cells level to brain function across different dimensions, across species, across the many modalities of cellular properties in healthy and diseased conditions, across space, looking at the whole brain and different regions of the brain together, and finally across time, from development to aging, and in different kinds of functional and diseased conditions. So with this tremendous information, what kind of questions, brain cell atlas question that can be tackled, greatly facilitated by using AI ML models. Here, I just give you some examples in terms of my understanding and also my wishes about what we can do. And as Stein beautifully shown, we start with single cell genomics data sets, and we can achieve to strive to achieve cross-modality data integration, knowledge expression, and new discovery, leveraging AI tools. For example, we want to relate or even integrate information at all these different levels: gene expression, chromatin regulation, spatial organization, temporal dynamics, and disease states of cell types across the entire brain. This is a daunting task. Large-scale computation will be needed, and we really hope that AI ML approaches will facilitate that. And then specific examples of use cases. We want to predict cell type identity, spatial location, developmental trajectory, and cell type specific enhancers. We want to define gene regulatory network, new anatomical postulation, new CCF, and cross-species conservation and divergence. We also want to predict changes, how the genomic transcriptomic landscape change upon perturbation of any of the components or disease variants and things like that. And by having this kind of framework, we hope that present to the community, allow users to flexibly extract any information about cell types in any modality and the relationship across different modalities. And finally, perform inference from the model using the new data generated by the community to compare and integrate with the reference data set that we present, and also transfer the knowledge into new data sets to make new predictions. Of course, then finally, beyond that, we further integrate with proteomics, with connectivity, and functional properties of brain cell types to achieve this kind of integration at the next level, combining genetic, structural, and functional information for the brain together. Okay, so this is like a gigantic wish list, from a biologist's point of view. Now I'll pass this along to Peter to see, what is the current state of AI. And I can advance slide for you.
Okay, thank you. So AI modeling for data generated by the Brain Cell Atlas spans several major areas. At the sequence level, as you heard from Steinert's keynote talk, sequence models are used to characterize regulatory activity from DNA sequence, understand the molecular effects of genetic variation and perturbation, and can uncover principles of the cis-regulatory code. At the cellular level, single-cell and multimodal foundation models are used to define cell identity, state, and lineage, characterize molecular responses to perturbations and environmental contexts, and integrate transcriptomics, epigenomic, and spatial measurements. At the tissue level, imaging-based models are used to quantify cellular and tissue morphology, spatial organization, and connectivity, and to relate molecular state to tissue scale structure. And I'll give you a high-level overview of some of these modeling approaches to set the stage for the panel discussion. Next, please. So I'll start with models that operate directly on genome sequence, building off of Stein's keynote. So sequence function models take DNA sequences as input and predict regulatory or molecular outputs, such as TF binding, chromatin accessibility, enhancer activity, or gene expression even. Conceptually, they learn a functional mapping from sequence to biological activity. These models capture regulatory structure across multiple scales, individual sequence motifs, how motifs interact, longer range interactions across regulatory elements as well. This makes it possible to ask questions that are difficult to address experimentally at scale. In practice, sequence function models are useful because they can help to interpret cis-regulatory mechanisms, predict the effects of non-coding variants, and increasingly to design novel sequences with desired activities. The broader space of sequence modeling is large, much larger than just sequence to function models. There are also generative models as well, including DNA language models and diffusion-based models. I didn't cover any of that right now, but if you're interested in this, if the panel is interested or other people have broader questions, feel free to ask it during the panel session. Next. So moving from sequence to cellular to context now, the next major class of AI models focuses on modeling cell state, particularly through single-cell transcriptomics. Foundation models in this space aim to learn general-purpose representations of cell identity from very large atlases, rather than being optimized for a single task, These models learn embeddings that can be reused across annotation, integration, perturbation analysis, and cross dataset alignment. These approaches build on other types of models, like latent variable models, such as VAEs, which are widely used to analyze individual datasets. Foundation models extend this paradigm by operating at a larger scale, emphasizing transferability and reuse across datasets and tasks. A major direction now is on bridging across modalities. The multimodal foundation models aim to integrate transcriptomics with other measurements, such as epigenomics, spatial transcriptomics, imaging, histology, all into a shared latent representation. And this allows different views of the same biological system to be aligned within a common representational space. Next, please. Next slide, please. Oh, that didn't turn out good. So beyond predictive and generative models, large language models introduce a different kind of AI capability. Here, yeah, thanks. Here, the LLM, a large language model, an LLM, is being used to interact with data, models, and analysis tools. In practice, what people call an AI agent is an LLM that can run code, call bioinformatics pipelines, query databases, read papers, and iterate across multiple steps. The two examples on this slide show how this idea is being explored. On the left, in Swanson et al., LLMs are organized into a small team with different roles, coordinated by an LLM acting as a PI, and the goal is to streamline the scientific workflow of antibody design. And on the right, with this very low-resolution image, apologies for that, Biomni uses an LLM as a front end to a curated set of biomedical tools and workflows. So the model can execute common research tasks, data analysis, database queries. It's a powerful way to leverage priors from human knowledge via scientific literature. And here I'm giving you a lot of these broad brushstrokes. Again, if you have any questions about specific areas of AI applications or want to change where the discussion leads, I encourage you to raise questions or comments throughout the panel. Next. So in today's panel, it's structured into 5 different sections. And the way we're going to go about this is we have lead panelists for each of these sections. And me as a moderator, I'm going to read a short setup just to establish the initial question for the lead panelists, who will each take turns answering the question. And then this will open up a broader discussion to the broader panel. So feel free to stay engaged. Unmute yourself if you want to contribute, raise your hand, and we'll call on you and have a broader discussion on these different topics. So the section topics we're going to cover are, where are we now, capabilities and readiness. Section 2 is on molecular and epigenomic data for functional inference. Section 3 is on benchmarks, evaluation, and CASP-like challenges. Section 4 is relating genes and cell types to brain function through multimodal integration. And Section 5 is on mechanistic interpretability and biological insight. And hopefully we'll have some time for some broader questions from the audience afterwards. Okay, so let's jump into section 1 because in the interest of time. So if Dave, Kelley, and Fabian Theus can, maybe show your videos so that we know you're here. I'll set up by, set up and ask the question, and please, I'll call on one of you to give the first answer. Okay, so AI ML models have rapidly expanded in scope and capability, especially for classification, annotation, and prediction tasks relevant to the brain cell atlas. At the same time, meaningful impact depends on how well these models align with biological questions, scalability constraints, and data realities. Understanding the current landscape helps identify which opportunities are ready now and which remain aspirational. So the first question I have for David, Kelley, and Fabian Theus is, where are we now in terms of development and use of AI models, and how can recent advances be leveraged to tackle brain cell atlas tasks such as classification and prediction? Dave, you want to go first?
Sure. Well, I feel like this is an easy one because Stein just gave us such a great introduction of the state of the art of what we can do with sequence-based modeling at this point. So, to summarize, I think these models are working very well. They are quite good at being able to predict local epigenetic marks and chromatin accessibility at a cell type specific level. And they're quite good at predicting gene expression evaluated across genes in a single tissue or cell type. But where we could still use some continued work is being able to predict, in this case, probably the most relevant would be cell type specific expression. So we've been pushing on this too with some brain data. And, the kind of easy things we can throw in up front are, wow, we can predict neuron specific versus microglia specific gene regulation. But actually, these cells are, quite different. You look at them and the differences are staring you in the face. Where we would love to be is to really deeply understand all of the subtle differential expression across different types of neurons, across regions, across different subtle functions that they might be performing. And then, of course, understanding how that changes in disease states. And, in a case like Parkinson's, where the neurons are dying off, can we really deeply understand what's going on there? So that's, I think, gives you a sense of where we're at and some of the big challenges that we're really pushing on. I guess just to briefly say, also to call back to Stein, I mean, the main work we do, the main thing I use this for are to predict the effect of genetic variants so that we can look at genome-wide association studies of neurodegenerative disorders. In my case, psychiatric traits, anything brain-related would be in play. And we'd love to have these models help us understand the genetics data to find map associations down to causal variants, target genes, and relevant cell types. And I would say that is in progress work that, we can pull out some anecdotes where it all strings together, but it's not something you can consistently rely upon at this point.
Oh, thanks. By the way, I apologize. I should have asked you to introduce yourself first. So maybe you can do that right now.
Sure. Mixed in there a little bit. So I'm a principal investigator at Calico Life Sciences. We study aging, neurodegeneration as a big push. We do a lot with genetics, human cohorts. And then, within my group here, we're bioinformatics, machine learning researchers. We build tools for modeling gene expression and other aspects of gene regulation, like epigenetics, as a function of the DNA sequence towards being able to produce these genetic variant effects.
Great. Thank you so much. Fabian, are you here?
Yeah. Hi, Peter. Thanks for hosting me. Can you hear me?
Yes.
Awesome. Yeah. I mean, I think David sort of covered quite a bit. Hey, good to see you, by the way. And Stein, of course, as well from sort of the sequence side. Maybe introduce myself first, so I don't forget this. Fabian Theis, I'm doing computational biology and sort of AI for single cell biology here at Munich, Helmholtz Munich. In particular, we've been doing some of the sort of standard softwares being used, such as ScanPy, and sort of building up this SCVerse ecosystem with a lot of friends, and a lot of sort of mapping, perturbation prediction type of tools. And I just want to start because, Peter and Hongkui, you guys asked about, where we are at the point with AI and ML, particularly for brain, but maybe more general cell atlasing, as we do also in the human cell atlas. And I think it's safe to say that for quite a few problems, it's already really operationally quite mature. This field has been sort of emerging very quickly. And I would just say even a few years ago, sort of data set integration and so on was a very big thing. But now so many approaches are out. And I think it's safe to say that, tasks such as cell type annotation, batch correction, modality alignment, but then also maybe even sort of predicting some readouts from other modalities, I would say in many cases no longer bottlenecks. Many of these really work robustly, quite a few particular scale, and also used quite routinely, maybe even in sort of standard analysis pipelines, or nowadays even in agents. And I think tools such as what we have with N8 and now scales to billions of cells. So even data sizes that are coming out now regularly. So there's like some experiments where you get 10 million cells at a time, right? And also, of course, spatial data sets sort of contribute majorly to that. And we can just sort of handle these robustly with particular deep learning tools. So I think this really goes massively beyond just sort of very basic things such as hierarchical clustering or so, what was shown before for marker genes. This sort of really mapping things in a joint space. And I think problems, of course, are oversimplification and sort of the biological knowledge in there. I think with nowadays Atlas level models, also foundation style representations, not just on the sequence level, but also on the cell and so on, I think this field has been really shifted to sort of more reusable abstractions. I think what's new maybe in the last 2 or 3 years, but relating to your question, is that I think it's maybe less now algorithmic major advances, but more sort of how we use that, share latent representations also across modalities. This transfer across data sets gets much more robust. So you maybe map something in joint space, maybe also not just across modalities, but maybe more from sort of an in vitro, let's say, organoid system towards a developmental system, maybe across species and so on. And you start interpreting these maps more and more. And that you can also train the models once and really reuse. And I think this across scales, from sequence to cell to tissues, is something that more and more of us are thinking about. But I think these sort of cross-scale embeddings are not entirely trivial. I mean, we've been dabbling quite a bit with sort of going from the cell to the tissue space by sort of working maybe on niche representations. We've been putting out this niche format as one example, and there's sort of many other type of ideas around how you sort of start mapping this in joint spaces. I think Stein mentioned a Scooby and related things where you go from sequence to also sort of cell type specific embeddings. And I think there's some of these ideas getting quite popular. But I think, and that'd be my last thing, like the hard part where this is going and where the limiting factors are not maybe so much a classification accuracy, but more its biological alignment, sort of, I would say models still struggle quite a bit with, what's causal, what's dynamic, particular, maybe sort of to summarize what's sort of interventional, like to really predict how cell state would change over time across perturbations or disease context. I would say it's more on the fragile side. And this is also where ground truth is not as much developed, because in the end, then it's not the cell number is not the limiting thing, but it's number of conditions, and that's of course drastically less, but of course getting scaled up now. So I think these multi-conditional embeddings, often what people now mean when they say virtual cells, these type of sort of generative perturbation models, I think is very exciting. And I think this opportunity that comes out of this is that you don't have the models in isolation, but that you can then put them maybe into loops, sort of learn from the data, guide next experiment. This is sort of the big hope. And I think some of these agents also, Peter, that you said in the end, sort of start moving towards that.
Yeah, thank you both. Your answers were so comprehensive. I was going to ask you what are the open challenges, but both of you have answered that question here. So I'm going to pose a little bit more challenging question. So right now, the state of AI in sequence modeling, in single cell modeling is quite maturing, right? And so there's no doubt that they're useful tools and they're here to stay. But as both of you guys mentioned, there are limitations, right? Because their ability to predict mutations or perturbations are still lagging far behind being able to, for instance, cell typing or just general gene expression. Sometimes it just works pretty well on average, but it doesn't really learn the nuances that well because it doesn't really understand a causal understanding. Now, a big proponent of this is that typically biologists generate data to study like questions or like, it's disconnected from asking what is the best data our AI models need to be trained on. So have you guys thought about this? And this would be a great platform to relay your thoughts, especially because we have so many data generators here on the call today. So maybe I'll ask David, why don't you go first?
Sure. So from the secrets perspective, I mentioned the frontier where we're just doing okay is being able to predict the differences between very similar cell types. I think there's an inherent limitation there. If you have two types of neurons and you do a careful RNA-seq experiment and there's 50 differentially expressed genes, then in some sense now my machine learning has 50 examples from which to learn this potentially very complex function, and that's just a tough place to be. And in other areas of machine learning, if your model doesn't work, you go collect more data. but I don't know how to go collect more differentially expressed genes if they don't exist. So I think we're going to have to get creative. Like certainly the first phase of this can be just making sure we're getting the best data we can to profile the cell states that we really care about. And all sorts of modalities can contribute to that. But from there, it's a little unclear. I'm personally interested in how we can share information across species. There are at least like lots of mammals out there, lots of different types of brains. And then other ideas might involve perturbing cells ourselves, which is probably a good transition point to toss it over to Fabian, who I think is thinking more about that type of modeling.
Yeah, thanks, David. If I might just take it up, I think it's a wonderful question, Peter, right? The end is like, what would be the next experiment? Or we sort of sometimes twist it around, and HC we also often asked, when are we done with the atlas or with the perturbation? So, what more do you need? And I think your point with the nuances is very well taken. Maybe taking a step back, I think what really made me and sort of, I think the world sort of really attend to early GPT and then later LLM models was that they were showing the scaling behavior, right? And I think it's sort of maybe common knowledge now that these scaling laws at some point showed, hey, now the model is sort of starting to pick up something interesting. And we see that it has been doing this from a smaller model. So if we sort of double the amount of data and compute, we get this and that much increase. And I think it's really important to say that our field, in particular the more foundational models, cell type of things, have not shown that yet. So it's not, I think it's far from clear. And I think this may be a bit different to the sequence models, David. Maybe there's some argument here that with these cell representations, these more tabular type of data, it's not clear that it's really this whole complexity of the model is entirely needed, right? So there's, on many cases, much simpler autoencoder type of approaches sort of outperforming some of the more complex ones. And of course, there's transition happening, but I would really like to see more and more of these scaling laws, like really downsampling the model, trying to see what's needed. And if you do this, of course, you can also say, what are the most informative features? And in particular, what type of new measurements do you need to take to maybe sort of lower down the overall uncertainty of the model, in particular perturbation regions and so on. So I think like how to reframe this in a statistical thing would be, what is a proper experimental design model? We early on did this with the junior PI here with Matthias Heinig. This was called SC power. Just a simple question. Do you need more cells or more transcripts for sort of maximizing some differential expression? And turns out that in this case, actually deeper sequencing didn't help. It was more cells. And for some other questions, you would have a different answer. But I think if we can answer some of these basic questions, we would know how to design the next experimental design. Like, do we need to distribute a certain perturbation budget, for me, for example, in neural organoids, on sort of more different pathways, maybe sort of more things within one pathway, maybe sort of genetic perturbations, maybe combinations. And I think it's safe to say we might need not more controlled PBMCs. But apart from that, it's not so clear. So I think having an actual model based on a certain data set, update the model, sort of do design of the next thing, I think that could sort of enable this question more robustly.
Thank you both. In the interest of time, I think we should move on to this section 2. Yeah. Okay.
Okay. Yeah. Thanks. So I will moderate section 2. It's more on the data side. It's molecular and epigenomic data for functional inference using models. The topic is very similar, to section 1, and David and Fabian have done, give us a lot of insights on this now. But we have a different lead panelists, different set of lead panelists. So we want to hear what they think about this as well. Our lead panelists are Jain Zhou, Mark Gerstein, and Jie Liu. I don't know who of you are here at a meeting. If you can show up, turn on your camera, that'd be great. Great, all of you are here. So I'll just read the setup questions and hear how you think about it. As I mentioned, the Brain Cell Atlas, we're generating rich molecular epigenomic data sets that capture gene regulation and cell states, creating opportunities to train models that can infer gene programs and estimate the function of genetic variants. But doing so effectively depends on appropriate data selection, labeling strategies, and modeling approaches. So the question is, how can this kind of molecular data types and AI models be used to infer gene programs and predict functional impact of genetic variants? And And then the follow-up would be that what data type is still missing that could actually great help accelerate the model building and improve our capabilities. So basically, what are the opportunities and what are the gaps now? Jen, you want to start?
Sure. So I'll start with introducing myself. I'm an assistant professor in University of Chicago, and I'm a computational biologist who's interested in understanding genomic sequences, their functions. So to talk about this question, I think when I think about the question of inferring gene programs from this multi-model data, which is very useful for understanding gene regulation in the brain and for the problem of predicting the functional impact of genetic variant. So I think of the question as understanding the causality relationship between the players of gene regulation, right? So what are the relationships between these DNA sequence and the various players, including the transit factors, the chromatin openness, the methylation, gene expression, and the 3D genome, right? So we now have all of this multimodal data measurement for those, but still a hard question and opportunity for us to understand what are the causal relationship among them and what is causing what, right? So, and I think this is a question that is good question to be tackled by a combination of computational modeling and the experimental approaches, right? From the computational modeling point of view, I think modeling, especially sequence-based models or sequence-to-function models, is very useful in dissecting the causality. So like from Stein's talk and Bing's talk, and many people here have first-hand experience with when we train a model to predict from doing sequence to these downstream activities, we can often capture this causal relationship, right? That will allow us to make causal predictions, like if you perturb a genetic variant, what's going to be the consequence? And if you use it to design a sequence, it's often having the activity that is can be experimentally confirmed. So I think that is an important power from modeling point of view to introduce the DNA sequence to help us better dissect the causal relationships between different modalities. You can potentially imagine that if we have a way to perturb a sequence to only affect A, and we can see whether it will also perturb B, right? Or you can have a way to perturb only B, whether it is going to affect A. So that's a potential hypothesis generation that we can make from modeling perspective. I don't think we're there yet, but I think that there's a potential to do that. And also there are many things desired from the modeling point of view. So we can make end-to-end predictions from sequence to, for example, chromatin accessibility or even to gene expression. That does not automatically tell us what is the process in between. So how does the sequence lead to these changes? Does it change it through affecting transit factor binding? Do they affect DNA methylation? And what is the processes that are leading to these changes? So one thing that's important is to interpret these models. And another thing I think is also going to be discussed in later of this panel, which is to build really mechanistically interpretable models where we might be able to explicitly model. So in the model, something represents, for example, transit factor binding, and some can represent histone modifications. And we can build these models that can potentially better help us understand this causal relationship, or at least propose viable hypotheses that can be validated experimentally. And of course, on experimental so computational modeling is not going to be perfect. There's not going to be always making correct predictions. So we really need experiment side to have data that help us understanding the causality in various type of perturbations, like it has been discussed already. It would be very useful. And also like even from the existing data, we have natural perturbations from genetic variants, like Stein has shown that we can obtain information from just looking at the allele imbalance within the data to have information about how genetic perturbation might affect the activities. So we have a lot of data in the brain cell atlas. Right? I don't know whether there is effort that we can do to actually identify the variants and cause allele imbalance in all those data types, that would be very, very useful for us to have a is there a validation, it can be a validation data set, or just giving us more information about how those variant or perturbations will have different effects in different individual cell types.
Okay, yeah, great. Thank you very much. Maybe next we'll have Mark. Would you like to give some comments? Yeah, your thoughts.
Sure. Can you hear me? Hello?
Yeah. Yes.
Okay, so I'm Mark Gerstein. I'm at Yale University, and I work in computational biology, and I'm very interested in the brain and the genome and stuff like that. So now, I think there's a number of questions for this panel. I'm going to sort of answer about, what types of things we need. And one of the things I would like to put out there, that one of the nice things we need is really population scale data. So, if we have this type of data on large numbers of people, one of the amazing things that we're always able to do is to use the natural variation in large numbers of people to help understand how the variants impact on the epigenome and how they relate to various functions. And this obviously has been demonstrated, throughout genetics from maybe the last 20 or 30 years. But I think it'd be really, really wonderful to have a lot of this type of single cell data really in population scale. And the second thing I would like to say is if we start to think about population scale, you probably all know there's a world of classical genetics that thinks about, QTLs and, all sorts of case control studies. And I do think it's really important to try to relate that world very well with the new AI models. Because I think that obviously that former world is fairly interpretable, but also I think it really speaks well to the how should one say, the sensitivity and power of these things. And one of the things that people have learned when looking at very large amounts of data, you can fool yourself very easily, and you can not replicate things and so forth. And I think it's it's good to really maybe put a lot of the AI models in conjunction with the kind of classical statistics to really get good confidence on how we're doing. And then the third point I would want to say is that in addition to a large amount of population scale data and so forth, we've heard about this notion of AI agents and having these agents go out to specific data sources and do particular calculations. And I think this is a wonderful thing and that we really want to do. But one of the things that really depends on is not so much the data, it depends on metadata, right? So we have to have our data sets really labeled well and all the conditions in the data sets, appropriately set out so the agents can figure out what they're doing to make them AI ready and so forth. And, so I think this idea of the small data, the metadata that's associated with our large data sets is a very, very important thing. And I think that's a great thing to focus on. And with that, I'll just end. Yeah.
Okay. Yeah. Great point. So population based on natural variation in populations, genetic variants, and then, yes, statistics and AI using AI engines. Yeah, great. Okay. And then Jure, can you share your thoughts?
Hi, can people hear me?
Yes.
Okay, thank you for confirming. So I'm Jure, Associate Professor of Computation Medicine and Bioinformatics, and also in Computer Science and Engineering at the University of Michigan and Ann Arbor. It's nice to see Jain and Mark's summary. I will not repeat that. Their comments are excellent. I'd like to add to the follow-up question, what data type is missing and what are missing there. I can see three components. I feel this is the trend at this moment. That is, from what I feel is kind of understudy in the Brain Initiative, this consortium. The first one is I see people build foundational AI models. I think a model will focus on what is available in the data set, right? I see in the last 10, 20 years, the sequencing data, DNA sequencing, RNA sequencing became so popular. Therefore, I see the model focus on these modalities more than other modalities. I can say, like, compare with protein and also RNA modifications, I think they are less studied. Therefore, they also reflect as when we build a model, we focus on DNA, RNA, transcriptome, epigenome, but we don't focus on too much on RNA modifications and also proteomics. But I can give one example. Proteomics is super important. I study type 1 diabetes a lot, and a lot of time when you look at the data from the patient, their insulin on the RNA level are the same, but it's really from pro-insulin to insulin, the transformation is kind of impaired. So therefore, if we only focus on RNA level, we don't really understand what's really going on there. So this just give one example. I feel the understudy part is not from the modeling itself, but it's really from data availability. The sequencing technology give us this focus, therefore we overlook other perspective. I think the next missing part I would like to emphasize is even now we have the joint profiling technology like single-cell multi-omic, we can profile RNA-seq, attack-seq for the same cell at the same time. But biologically, when the cell during a development or disease status changes, these two modalities may not synchronize. A lot of time, maybe the epigenome maybe precede the transcriptome. So I feel when people build foundation model, they just throw in the two modalities from the same cell, and they assume they can model in this way, but there's always maybe some implicit bias introduced in this type of modeling. Even the two modalities are profiled for the same cell. I feel people should pay more attention and think carefully what's really going on, and maybe do a more careful way of jointly modeling, building the true biological, truthful multimodal AI. And the third one, I feel probably people also aware is the emerging of spatial technology, no matter it's for spatial proteomics or spatial transcriptomics. I feel all the tissues, the neighborhood play a role. And if we're only looking at the RNA-seq, attack-seq, without knowing the location, I assume the human brain also, the tissue also has the regions, and maybe there, at least I know the human brain has multiple regions. I feel having this location information will complement the existing single-cell technology, and probably integrating spatial information in the modeling will, I will say, improve the understanding of the disease. Okay, I think that's the three missing part I see in the field. Thank you.
Yeah, very good point. Very good point. Missing data types, consistency across modalities, spatial information, and also, very consistent to what Jen was saying. It's not just the major data modalities we're working with, but other kind of modalities that can bridge the relationship, so that you understand, and also the natural variation. Basically, from data, you understand better the relationships and begin to really gain mechanistic insights. Things when different types of data come together, being integrated, then things become more make sense, and you can make better sense of the data, and the models can make better sense of the data as well. Yeah, very good point. Okay, so again, thank you very much. I think in the interest of time, we need to move on to the next session as well. Hopefully, this section panel will end from the NIH instruction. It will end at 4:35 p.m. Eastern time. So we still have some time, but we'll move on with other sections. And if we have time, in the end, we'll come back, do an overall synthesis as well. So, but let's move on, Peter.
Okay, so section 3, this next one is on benchmarks, evaluation, and CAFS-like challenges. And the lead panelists for this section are Žiga, Avsec, Rahul, Satija, and Yusuf Roohani. Are you guys on? All right, so I'll just get started then. Progress in AI ML is accelerated when the community shares evaluation standards, benchmark data sets, and common performance criteria. In other domains, challenge-driven frameworks such as CASP, ImageNet have really helped define goals, expose limitations, and guide innovation. establishing similar frameworks for the Brain Cell Atlas could really strengthen the rigor and comparability across methods. So the question is, how should we define objective benchmarks for evaluating AI ML models for the Brain Cell Atlas? And remember, first introduce yourself, your position, and then you can go into answering that question. So I'll start with Žiga because I already know you have lots of opinions on this topic.
Great. Hi, everyone. Good to be here and good to hear to others about their thoughts. I lead the genomics team at Google DeepMind, and we're very interested in developing models that will help us decipher the genome sequence. Our most recent model is AlphaGenome. And yeah, I have lots of thoughts on evaluations. Evaluations are things that is not as glamorous and that, is not as exciting, but it's really, really important because it really sets the target of what we're trying to hit, and it sets the target for the whole community. And that makes it, really, really critical. So sometimes good benchmarks are even more important and more durable contributions than the next version of the model. In terms of how to define benchmarks, I think it's really critical to be very clear about what we're trying to achieve with these models. For sequence to function models in particular, as David and John and others have mentioned, and Stein have mentioned, it's the one important task is the interpretation of genetic variants. And there we really care about causal understanding of these models and the zero-shot generalization of these models. So without training on these data sets on variants, can we generalize to them? And there, it's really important to have causal perturbational evaluations. Stein showed some of that with these allele-specific changes or EQTLs. But other things like CRISPR perturbations are also really, really important there. One thing, in terms of CAS-like challenges, one thing I wanted to highlight here that is quite hard sometimes to pin down a single number that you're trying to optimize, because there are so many different biology is complex, and I feel like protein structure prediction is a simple task compared to trying to understand, how the brain works, for example, right? So, and because there's so many different aspects of it. So trying to codify as many useful metrics as possible is always helpful to get like a balanced view. So not just measuring things in a single cell type, but across also other types of cell types, for example, with different technologies, because different technologies have different biases and you can achieve different kinds of scales. And it's also important to have sufficient amount of data to get a small, to kind of reduce the variance of evaluations, because so that you can then compare models to one another. The other thing I wanted to say is that it's also helpful to have a different ladder of evaluation metrics where it's not just about the end goal. So for example, for functional variant interpretation, you want to find map, say, GWAS variants. But you want also upstream metrics so that you can kind of understand and make progress on simpler tasks first, so you can walk before you can run, essentially. And I think that's really critical, because sometimes some of the problems are stiff, right? And it can go for years or tens of years or a decade when you're not making progress on those, but you're making progress on a simpler thing. And I think that's really important as well, and it allows you to also bisect and focus your effort on improving this. So and finally, I think it's really important that we share these benchmarks as a community and also curate. So for this consortium, I think it would be really helpful to provide some of the such as like the files, the targets of what you're trying to predict in simple like CSV files or something. I think that's a more important start and the key metrics than organizing a challenge, because challenges initially, they can be gamed. So I think it's better to start with more metrics that are easy shareable, that people reuse in papers. And then as things crystallize, as you start to see which are the key benchmarks that are reliable, then you can start to organize challenges and get clarity. So I feel like it's more of like a funnel approach where you have to time it right as when you start to focus on a single number, because it can be dangerous if you do this prematurely.
Great, thank you. Rahul?
All right, hi everyone. My name is Rahul. I'm a core faculty member at the New York Genome Center, and my lab focuses a lot on developing tools and technologies for single cell analysis. I did a session earlier today on integration and annotation, which is what many of you may know my work for, but actually my group over the last couple of years has really changed our focus to work quite a lot on understanding single cell perturbation technologies, both computationally and experimentally. So we think a lot about some of these challenges that people are running. And one of the things that I think is always helpful to highlight to the community generally, we think a lot about AI and biology, but there are very different subfields of this problem and different types of things that people work on, which in my opinion, at least, and I'm curious what Žiga thinks, are at vastly different levels of maturity and really change the way that we can or should be using benchmarks. So I'll walk you through kind of how I see the spectrum of this. On one hand, we have something like protein structure prediction and AlphaFold, and I would argue that the benchmarks and metrics that Google and others developed for this were essential in the incredible maturity that field has been able to establish. People use AlphaFold and they trust AlphaFold because they understand the metrics. They know that when a method achieves a certain metric, they know what that means, and they know that can achieve a certain level of trust in the computational predictions. Also, it was trained, of course, on gold standard, human understood, highly annotated data. And I think that is, in my opinion, an example of a very highly mature benchmark-ready field that's had a tremendous impact. Something more in the middle, something like cell annotation integration. Obviously, those fields are significantly more mature than they were, a few years ago. There's no doubt about that. There's still some question on how good the metrics are, I would argue. We don't necessarily have ground truth a lot of the time on what is a good integrated data set and what is a good cell type annotation. We spend a lot of time in HubMap trying to generate sort of real ground truth, or at least not ground truth, but at least enough people have okayed it to be treated as ground truth kind of data that we can use for this. But that's an intermediate problem where I wouldn't necessarily say because something achieves a certain benchmark, I trust it, but at least I have a reasonable sense of what the benchmarks mean, and that's an area of further development. And then if we get to the other end of the spectrum, we have something like cellular perturbation prediction. And this is tough. This is a new field. The data is noisy. We don't have nearly enough of it. And so on one hand, of course, it's really critical that we have quantitative, benchmarks and metrics that we can use to optimize these models. On the other hand, I would argue, we don't really know as a community yet what those should be, and they're very easy to game. So to give an example, if you're trying to predict the effect of a new perturbation and you're able to predict all the ribosomal genes right and none of the other genes, you're probably going to get a lot better of a value on most benchmarks, whether it's R squared or energy distance or things like this, because those genes are so highly expressed and there's so many of them. But from a biologist perspective, that's not necessarily what we're trying to do. We don't want to understand structural proteins. We want to understand, more complex and variable biology. But the metrics yet haven't been able to catch up with that, at least in my opinion. So that's an area where, just because a method achieves a certain metric value, I don't necessarily know as a biologist what I feel comfortable, using it in my own lab, or would I even necessarily feel comfortable saying one method is better than another because it achieves a certain metric. Doesn't mean we shouldn't do it. It just means we have to be aware as a community of what some of the challenges are there. So none of these have easy solutions, but I do like to highlight the sort of range of tasks and maturities, because it's very easy to loop all of this together into one lump. But they are very, very different problems with different types of data, different metrics, and different levels of annotation and training. So I like to highlight that.
I think that was really well said. And, I think both of you have highlighted really important points that, on the elegant, on the easy side, CASP seems like a well-defined, easy problem, right? The metrics itself are aligned with biology. You know exactly what you're getting out when you hit a certain target. But in these other areas of biology, these other prediction tasks, we don't really know what that is. So going back to that point, Rahul, you already mentioned some of the challenges in single cell field. Žiga, do you think that the community is ready for sequence-based models? Because I guess genomic perturbations, you kind of have what the readout is, right? It's just gene expression. But I was just wondering what your thoughts are on whether you think the field is mature enough to move forward with a CASP-like competition.
Yeah, that's an interesting question. I'm kind of flipping back and forth on this question. So I don't have like a strong answer to this, but basically. I think on the one hand, yes. On the other hand, it would be helpful to have even more of these evaluation data sets out there. And also, like, I don't think, I think, basically I'm kind of in between. Maybe for some of these benchmarks, it might be helpful. But we have to be careful with what, just because something is easier to measure and then define a challenge around it, that doesn't mean that it's more important to tackle as a community. So we also have to be careful about that, basically. So for example, MPRAs are a wonderful technology and great, but they don't measure everything that we want. So and they might be the easier technology to start challenges, but you might still be miss a lot of these other things. So that's why I think perhaps a repository of all the useful data sets and benchmarks are out there, curated well for sequence to function models would be a better start, in my opinion. And then from that onwards, you could then add these challenges as a bonus, basically. So I would sort of order it in a different way.
I tend to agree with your assessment in that a lot of papers, they create their own benchmarks and they're always soda on their own benchmarks, right? That's fantastic for you, but when people try to replicate that study, it's always often hard to like download someone else's benchmark data sets, reprocess it. Is it processed the same way? If all of that was standardized and uniform, this would greatly help the community out, I'm sure, in every subfield of biology. Rahul, do you have any opinions on like, I guess, there are some points about aligning, the tasks or the benchmarks with something that a biologist really cares about. Like, do you have any opinions on like how we can navigate that or move forward?
Yeah, it's really, so what would be a metric that really captures whether or not the model predicted the true underlying biological function of a perturbation outside of its R-square value? I don't have an easy answer to that. I like to draw an analogy, though, in terms of as we as biologists, and I have real biology projects in my lab, so we think about it from a user perspective. When would I feel comfortable not doing a screen, for example, and just using a sort of AI model to replicate that experiment? And that's a question more about trust and about sort of belief in the biological abilities of something more than achieving a certain metric. And the analogy that I like to use is, many of you now do single-cell RNA-seq in your lab, and you were probably at some point choosing between many technologies that you might want to use. And some of you might have run multiple technologies, but I doubt many of you sort of simply looked at benchmarking papers and took a metric and said, okay, this method has the best benchmark, so I'm going to use that one. You probably talked to your colleagues, or you looked at some data, or you may have used the benchmarks in concert with other pieces of information, both anecdotal and otherwise, that together sort of led you towards a decision. Like you might have seen a colleague publish a paper with the result that you're like, I want to be able to do something like that. So I actually think as important as benchmarks and challenges and structures are, and Yusuf's not here, but the challenges that they've put together have really engendered an enormous interest in the community. Brought a lot of people together, they've had enormous good. But I don't necessarily think that the winner of that challenge is something that we would just say, okay, let's all use this method. I think that's looking at it the wrong way. I think we should be okay as a community in saying, we don't have to only go on quantitative metrics to kind of figure out what's working here and what's not. And all of the sort of different things that allow us to build trust as we see real methods discover biology that we believe and replicates and validates. Slowly over time, we build trust in things. And that's, I think, what happened with AlphaFold as well. So we're thinking a lot about, how do we help build that sort of trust? How do we sort of help make that process a little bit faster, so that methods that really are more mature, people can kind of use them. And part of that is making them easy to use and making data easy to get their hands on. But to me, it really is important to highlight the limitations of metrics in that type of decision-making process as well.
Absolutely. I guess to bridge both of your thoughts here, some of the downstream biological discoveries using these tools is a nice way to actually show that those tools that were useful, and that could be a part of a larger collection of evaluations to simulate whether your method could have come up with the same conclusions, at least because those are vetted and validated much more carefully. So sorry, I apologize, but we're out of time for this section because we have 2 more to go and we're almost out of time. So I thank you very much, but we're going to move on to section 4 on relating genes and cell types to brain function through multimodal integration. And Hongkui, are you here?
Yeah, I'm here.
Okay, your screen is frozen, okay.
Really? Okay.
Take over.
Yeah, I don't see I can see myself moving, so hopefully that's right. So we for this multimodal integration session, We have some lead panelists as well, but I'm not sure who are still here. We have Reza, Abbasi-Asl, and Trey, Ideker, and Viren, Jain. I know.
Hey, Hongkui, I'm here. Reza.
Great, Reza, you're here.
Yeah,
Okay.
Trey's here as well
Trey,
in one video.
Trey's here as well. Okay. Okay, great. So just quickly, talk about the question itself. As you know well, to understand cell type-based function, it's critical to examine and integrate the different modalities of cellular properties together. And now we're going beyond just omics, including transcriptomics, epigenetics, spatial, proteomics, morphology, connectivity, physiology, and function, all these things. And this kind of integration would allow us to uncover how different cellular properties change in a coordinated manner, right, with time and space and under different conditions, functional or disease conditions. So the question is, how can we link the different data modalities together, molecular, anatomical, and functional, across space and time to reveal the dynamics and heterogeneous cell states? And what are the challenges? What are the possible solutions? Maybe we can start with that. Reza, you want to go first?
Sure. Hey, everyone. I'm Reza. I'm an associate professor of neurology and bioengineering here at UCSF. I'm a computational neuroscientist and a computer scientist by training. My primary research interest is actually relevant to the question. I'm really interested in using AI, advanced ML, to integrate across modalities. Now, that includes molecular, cellular data, but also in another level, more macroscopic data, just get characterization of function to understand the relationships. And I think maybe to sort of my take on the question and the sort of the angle that I would like to highlight is maybe from two different perspectives, from the modeling side and from sort of the functional characterization side. On the functional side, on the modeling side, of course, as others have highlighted, there are lots of progress in the field of AI. There are studies out there that demonstrate sort of translation of and utility of some of these tools in biology. But I think, in my opinion, going forward, there are a couple of maybe dimensions that are are still maybe open question. There are questions about those, and especially when it comes to multimodal integration. Many of us maybe even start with off-the-shelf models, but when it comes to utility in biology, at least our experience has been that there needs to be modifications to tune models to target modalities that are of interest to us, right? So just using blindly tools out there could be misleading, although maybe performant. But when it comes to extracting insight or getting that biological insight, there are many examples that things could go wrong if you're not really careful about the assumptions behind the model. So I do think that there is a need to start from scratch, same foundations, same principles that are out there in the computational side, but I think there are additional considerations that need to be included. Like, for example, when we train a model, needs to be there. Just one example of that, I would say, from work in our lab has been, we've been really interested in neuroanatomy and how we can use molecular data to come up with sort of brain mapping and brain parcellation. And we started with just off-the-shelf models. Things work, but we realized that there are lots of sort of inaccuracies that are due to assumptions in the way that the model originally was trained. So we had to actually come up with a new training schema to model neighborhoods in sort of a cellular tissue in a tissue and within a cellular organization. So I think that is like a good for us a good lesson learned. Kind of going to maybe thinking about a question of integration. I think harmonization has been a big issue, whether it's across modalities or in the space. So there are resources, of course, Common Coordinate Framework from our institute that helps that integration in the spatial context. Still, there are lots of different data sets, even like within same modalities, data coming from different devices may need sort of additional harmonization. Now, this sort of new wave of computational tools that are like foundation model level approaches to some extent address the need to harmonization, but I think there are still lots of questions there. We don't know necessarily if that's like fully addressing this question. So I think that has to become again a key question to ask. And then one final piece on the modeling side is like how we relate actually a model trained or observations from a model to the function, right? So that sort of biological insight, how to extract it from the model, it's still an open question. Sometimes you directly model it, but sometimes you have to infer it from the trained model. So that's still, to me, an open question. On more like functional side, I guess there are lots of questions. There are biologists on the panel that I think are more expert. My lab, we are mostly interested in functional or sort of characterization in spatial dimension and in temporal/longitudinal dimension, which kind of goes back to the question of parcellation itself. I think now an open biology question, whether we should stick to more traditional sort of way of neuroanatomy, like drawing borders on images, or with this sort of rich, new, large scale, massive data sets in molecular scale. Do we need to rethink the way that we define functional organization in the brain in a spatial resolved manner? I think that's also a really interesting question. My opinion is like we have to shift. There might be even things that are more probabilistic rather than like a sharp border. So I would love to also hear others' thoughts on this, but that's where we are, I think, heading as a sort of as a community.
Okay, great. A lot of good points. We'll get back to you. So Trey.
Yeah, thanks, Hongkui, and also for the invitation. So I think the reason why Hongkui invited me is one of the reasons maybe is I'm PI of a Bridge to AI Common Fund project called Cell Maps for Artificial Intelligence, where we're this section is on multimodal data integration, and in that project we're doing it's really focused on how you would build a foundation model of cell architecture. That is to say, what are all the components inside of a human cell, and how do we map and model them structurally and functionally through integration of spatial proteomics, regular proteomics, and Perturb-seq style data. And there we also have, our flagship model is a human iPSC, and that's all on our website and has been used in some of the challenges that were raised in the last section on benchmarking. But we're forthcoming data, not yet produced, but we're working on it, are NGN2 differentiated neurons, NPCs and neurons. And so that'll be, I think, exciting for this community as a model of subcell architecture regarding, neuropsychiatric disease. In terms of, I just have two, I can make this real short. In terms of what are the challenges of multimodal integration, I want to raise two things. One is missingness, and the second is interpretation. That I think, I think Hongkui, you've already pointed out what are the advantages, as well as Reza have pointed out the advantages of multimodal integration. So the first one on missingness, if you can imagine, if you have this many genes or proteins in modality 1, this other set of genes and proteins in modality 2, another set in 3 and 4, pretty soon that Venn diagram is very small in terms of the intersection of all of data where you have, or a set of proteins where you have all data types measured. And so if you have to always take the intersection of data that are being modeled, which is the assumption of your out of the box, multimodal self-supervised learning approaches these days, you're going to have a decreasingly, a decreasing set of proteins that are actually mapped in all experimental data sets. So we definitely need approaches that allow for interpolation and missingness in terms of the data covered and can approach more like the union of molecules rather than the intersection. That's point one. Point two is interpretation, which I think is the whole next section. So I'll just keep that real short. Obviously, the more modalities you have, it gets really confusing. Why did a gene or protein or cell, or in our case, protein complex or organelle, whatever you're embedding, right? Patient, why is that object embedded at that X, Y, Z, W coordinate? If you have one modality, that's an easier interpretation task than if you have 7. What is the exact, because every embedding is going to, of a particular data object is going to have some attribution in terms of which of those modalities were more important versus less important. We don't currently in the field have great methodology for that, but I think a lot of good people are working on it. But maybe that's a segue to section 5. So that's all I have to say.
Okay. Yeah, very good point. But I think the point you raised is really, really critical. Multi-model integration, if the data are not completely coherent with each other, if they are, the intersection space becomes smaller and smaller because they are scattered, then you really don't, get much information out of it, right?
We've been having this exact problem with our,
yeah. I mean, the cell architecture versus brain architecture, same thing, but also unbiased, or not biased data types, right? You have millions of cells in one modality, but not so much data in the other modality. It's not going to give you good integration either. So then what do? Do you wait for more data to become available? What can you do facing this problem?
There's machine learning methodology that can handle it. I think it's totally expected that you're going to have imbalance across your modalities in that way. And there are approaches, that are beginning to address this. So I, it's just people need to be aware of the issue.
Yeah, just maybe to add to that,
and the correspondence is really important. In the Venn diagram, you really want things to actually correlate with each other. You want to make the intersection as big as possible. Yeah.
Well, that in terms of the incoming data, but then wherever the incoming data leave you, you want to push out towards the union if you can, and just handle the fact that, maybe you don't have this proteomic layer or this perturbation transcriptomic readout for that set of genes. But maybe you can, back to your the whole prediction discussion you guys just had, maybe you can tell the user what that data set would have looked like had it been included and interpolated from the other 3 modalities you do have.
Yeah, just to add to that, from the computational perspective, I guess, as you said, there are tricks to do interpolation. Just last week, actually, there was a really nice paper in Nature Methods on how you can actually do dense sampling of spatial transcriptomics sort of slices just computationally in silico. But also there are tricks on the modeling side that now these days try to address some of these sort of difficulties in missing data component, like techniques from like few-shot learning. Once you have like a model trained on one modality, but the other modality hasn't, it maybe is not as comprehensive, what would be a good sort of training mechanism for the model to account for that? So I feel like there is actually progress on both sides, but still needs to be explored. So we have a long way to actually be able to meaningfully utilize that within biology.
Yeah. Okay. Yeah, really great points you two have raised. So let's move on. We have 14 minutes left. Actually, I know everything converges into this final section about the mechanistic interpretation and biological insight. So Peter,
right.
Please discussion.
Yeah. Thank you. So the lead panelists are Stein, Aerts, Jingyi Li, and Sudipta Sengupta. If you're here, please, share your video. So again, this is the final section. We don't have that much time, so I'll just get straight to the point with the question. I'm going to ask the first question to Jingyi and Sudipta because we've already heard model interpretation through Stein, but I will follow up with a more difficult question for Stein after these first two questions, okay? So how can we, design AI ML methods or develop, post-hoc interpretation methods that move beyond just relying on, prediction on held out data to support a deeper mechanistic understanding and gain biological insight. So Jingyi, why don't you start?
So I know that interpretable AI or explainable AI is a very popular and important field, right? So the question here is what level of the metrics people design, because the interpretability is itself a vague term. How do we define that? And whether we can use that to connect with biology, and can we use existing knowledge to basically constrain the interpretation? I think these are all very important machine learning AI research questions. And also beyond that, I remember there was a very recent archive paper, I think from Ginevra Allen, who's at Columbia Statistics Department, which basically mentioned that stability and interpretability are different things, or basic question whether we can use stability, which is something we can observe, to guide our interpretability of the model. So I feel like there's a lot of questions to ask, and maybe it will be easier to ask this question with a specific task and model rather than a general discussion. That is my feeling.
Yeah. Thanks. That's really nice. Sudipta, do you have anything?
Yeah. Hi. Let me quickly introduce myself. I'm in Amazon AWS, working as vice president and distinguished scientist. I'm also working closely with the Allen Institute, Hongkui, and Shoaib on the call on multimodal foundation models for single-cell transcriptomics. I want to first size the problem. We talk a lot about explainability and interpretability. A lot of that means feature explainability, right? Which features in the input lead to the specific output of the model. But in this case, when I read the title of this part of the panel, it's about mechanistic interpretability. And the key part is the word mechanistic. It is about how the model thinks internally. So the bar is higher. The expectation, the detail of understanding is more than feature level importance. So if I can quickly share my screen, I want to show you some papers and websites, but let me frame where I'm going at. So this is a very active area within the computer science field with large language models, mechanistically interpreting them. And there is some recent work. Let me quickly try to share here.
So you're referring to sparse autoencoders? Or
yes, I'll come to that in a moment. I'll come to that in a moment. Can you see my screen?
Yes.
Yes.
Yeah. So this is a relatively recent paper from Anthropic talking about how to interpret how these models think internally. It's not in the context of biology, but let me frame it because these models, as you know, are based on transformers, the transformer architecture. And a lot of the biology models today are also using transformer architecture. So anything we can do with transformers, we can try to combine with concepts from the biology domain and carry over the same techniques. So this blog post is backed by 2 papers, which you all can go and read if you're interested. But I want to quickly highlight one example. Here you can see it is generating a rhyme. These 2 lines. He saw a carrot and had to grab it. His hunger was like a starving rabbit. So the question is, if I am just after grab it and predicting the next token, is the model planning ahead, or it is just thinking about the next token, which is his, right? That is a fundamental question about what is happening internally. And so one of the standard techniques here is to look at the internal activations of the model. Again, for the biology community here, an activation is just a vector of numbers. It is relatively opaque, and that's where the difficulty comes from. But you can try to intersect it with known representations of this model for rabbit. And what the authors here found here, that the internal state at this point indeed has an overlap with rabbit. So the model is thinking ahead, planning ahead, because the second line has to semantically continue from the first line and also rhyme with it, right? So that's kind of the thinking, the state of the art that we can like we have today in our toolbox to kind of mechanistically interpret what is happening inside these models. There is another interesting chart here. I'll not go over it, but how it is adding two numbers. Interestingly, it's not following the carryover rule for adding two-digit numbers, but it is using some approximation for adding 30 and 50 and then fixing the units digit and so on. So that's kind of the broad setting here. And then let me now switch to the biology domain. Some of you might know about the EVO2 embedding model developed at Arc Institute. We are actually using it in a Bloom model, being developed at Allen Institute for encoding a gene sequence. So the question is, this model does just embedding, right? But there is a lot of knowledge built inside the model through the training process. And not all of it is visible. A lot of it is latent because we have constrained the output interface of the model. So can you look inside the model and try to understand what other intuition it is learning beyond what it is outputting, right? So interestingly, there is a paper very new of the press from this company called goodfire.ai, and they are interpreting EVO2 to figure out what kind of biology it is learning and how it is coming up with the answer. And so let me quickly mention what it is doing here, because sparse autoencoders are mentioned. So it is essentially the techniques here found that the model, if you can train a separate sparse autoencoder model to analyze specific layers of the EVO2 model, and in this case, they looked at layer number 26. It's one of the later stages of the model where the semantic meaning comes out. They found that the model, again, analyzing the features internal to the model, they found that the model is learning coding sequences, the parts of the DNA that contain instructions for building proteins. It is also learning protein secondary structure, the way proteins fold into alpha helixes and beta sheets, and other things like the RNA molecules involved in protein synthesis. And really the way, the one diagram here that says it all, they use this SAE model to figure out, give meaning to the activation. So you can see this diagram here where there is blue color coding for activations which correspond to alpha helix, then yellow for beta sheets, and dark blue for tRNA. And they have overlaid it to the alpha folds output of this particular gene sequence, and they correlate really well. So even though we are training EVO2 to just do this sequence prediction using autoregressive modeling and also outputting and embedding, there is so much of latent learning going on that is coming out through interpretability. So just to summarize here, I think there is a lot of progress happening in the large language model world on transformer architectures. And if we can intersect some of those techniques and develop new ones for the biology domain, there is a lot of promise here to uncover the hidden knowledge that these models are picking up. And in this case, it's all from unsupervised data. It's just the gene sequences in EVO2.
Thank you. But we have to move on to give Stein some chance to talk because we only have 3 minutes left. All right, so with mechanistic interpretability, they actually, there are people working in biology already doing this stuff, but it's still just a correlation analysis. So I think that there still is some problems where we don't actually get out mechanistic understanding from them. So now to shift over to Stein, what key bottlenecks do you think do we face in model interpretability, and what promising directions could help overcome them?
Let's think. Well, first of all, I think it's useful to categorize a little bit in terms of cell identity and the atlases links the levels of the individual enhancers and regulatory sequences, the level of gene expression and the level of gene regulatory networks. Because at the enhancer level, there is a lot of progress I think we can get from these models. And I think at that point, we are at the point where we can actually start thinking about rules, grammar, and mechanistic models that may allow us to generalize better compared to the black box models, which at the end of the day are fitting well on the data if we are lucky, but we don't know if it can help us understand the edge cases like David alluded to. Sometimes there's not enough examples of co-expressed genes or cell type specific enhancers. Particularly if we look at development, there are transition points where there may be, singleton enhancers, idiosyncratic enhancers that integrate cues from patterning, of which only one example exists in the genome. So either it's going to be across species, but then many species, and this is going to be hard, or we have to really get to a mechanistic model like we were doing before the CNNs arrived, actually. And then we got into this amazing revolution. And maybe I think these models will help us to train the mechanistic or probabilistic models better. Think about having rules that you can test through design, which never worked before the CNN, but now they should start working again. We are trying these things out where we think we understand how an enhancer works, design it by the mechanistic model, have it first analyzed by the black box model, and then test it also in vivo. And this way, hopefully we can get to this interpretation. And the next two levels are much more problematic, I think. David Kelley already alluded to at the gene level, the way enhancers cooperate, these rules are still a little bit less clear, I would say. And so there, we first need, I think, progress on the black box models, more progress there, to reach the level that we have on the individual small sequences, and then think about the mechanistic models. And at the gene regulatory network level, it's to me even more problematic. And we need that to get a mechanistic understanding. Viren alluded to it. The gene regulatory network will be a very nice way to get a mechanistic understanding and to work on the dynamics, as well as interpretation of cues and cell states. The connection of the trans environment with the cis environment are needed. So thinking about better data sets for ground truth and perturbations that are really fine-tuned to these models, not just blindly generating perturbations, but how these things go together, the modeling and the experimental perturbations, I think would be really useful to get the GRN models to work, if that helps.
Yeah. Jessica, because it's been a while since you said anything, I just want to do a quick short question for you. So a lot of our current model interpretation tools, including SAEs, are just learning correlations. We're trying to interpret our model, but they're even correlations according to the worldview of the model. Now, coming from a statistics background, there are tools to uncover causality. And so if you assume that the world model, according to the model, you can query the model however you want, there might be tools to leverage from statistics. Have you thought about this, or have you had conversations with people? And so maybe you can give me your quick thoughts on how to bridge that gap.
Yeah, very, very good question. So there's a field called causal discovery that exactly does what you just described, right? How do we learn from associations to infer a possible causal graph, like which variable causes which? But I think there are some necessary assumptions we have to make. And so without the assumptions, we can never go to the causal conclusions. And I feel like what's missing here in our community is that these assumptions are usually not specified, or how they are related to our biological knowledge, or whether there are assumptions that are reasonable or not reasonable. I think these are hard. And also, given the number of variables we have, so high dimensional, that's another challenge. Do we really want to first screen down to a set of key variables we care about, and then talk about causal relationships of those instead of using everything? That may be a feasible direction as well. And once more, I feel like the data measurement noise from a statistical perspective is another thing we need to consider, because we can always pre-process our data to change the data quality, and that could also change the modeling and also the conclusion and how that will play a role. So these are all the open questions, but I do feel that it's very important. And one more thing I want to add that is, in my opinion, or in the statistical language, causal relationships and mechanistic model are also different, because causal means that the effect is at a population scale, right? It's not like for every cell you perturb it, you see something, but it's like if you look at all the perturbed cells versus the unperturbed cells, you see a difference. That is causal. But mechanistic thinking, like in terms of physics, that's even stronger. You want to see something more like deterministic, right? If you change something, then something must happen.
Mm-hmm. So in some sense, maybe causal like explanations or causal interpretation might be a good way to get some broader insights. And the mechanistic understanding is actually a much harder task. So that is a second follow-up of what we should be aiming for. I guess. Okay, thank you so much.
Thank you.
We're all out of time. I think I want to thank all of the panelists from all the different sections. Hongkui, do you have anything to say? Add on?
Yeah, I don't. I just wanted to say it's really fantastic. We have so many AI experts here today. Everybody gave insight. I learned so much today. So I hope our audience here, BICAN members here also learn from all these experts. So I just want to express my appreciation. It's a really very stimulating session, and thank you very much.